Thank you, Roger, again. It's a pleasure to be here and talk to you all about some of the work that we're doing in terms of gene discovery and validation and trying to really stay on track with the discovery process, which can be tricky. So the reason why we're doing this, obviously, it's important and it's the driver for us. Gliomas are quite terrible. They are unfortunately quite deadly to most people. Survive. The median survival is about 14 months. As you can imagine, it's not just about the deadly survival rate, it's also about following them up, these patients, and having access to tissue is quite a challenge, and even more challenging, the following up of these patients with access to tissue. So there are a lot of questions to be answered, a lot of work to be done in this field for sure. So I thought I would give two examples of two studies that we've been doing, and with a special emphasis on the validation process. And it's not obviously perfect, but it's just a hint of some of the work that we try to do to validate what we're doing. So the first study is a comparison, really. This started with a comparison of known methods and some homemade methods to perform RNA sequencing. So this one particularly is comparing total RNA sequencing versus two methods that enrich based on exome regions. And in this case, we've used glioblastoma samples, and specifically it's plasma from these patients, and their exosomes isolated with a kit. And the first study was done using 8 ml of patient samples, with about 2 ml typically for all samples. So the first cohort was 2 females, 6 male patients, and we tried to get the control samples to match the patients as much as possible, as you can see here. It's not always possible, but in the small cohort, we were able to do that. So it's quite well balanced between sex and age. So this is the overview. So as I said, the method 1 will just involve total RNA sequencing. And the remaining 2 methods are home-based methods or improved methods to really try and understand how hybrid capture works in this context. So this is an overview of the QC. So total sequences versus method 4 and method 5. So you can see quite different reads, that mapped reads of these of the captured genes. And at first glance, obviously, you can notice that the hybrid captures work better for these, and they're quite outperforming the total RNA sequencing method. If you look more specifically at the biotype distribution, again, you can see that with method 4 and 5, all of them include a ribodepletion step. But even with that, with method 4 and 5, you're able to detect even a better mapping of the coding regions, as you can see down here. So I'm just trying to manage. So in terms of the sample analysis, you get the genes, you get about 80% of better gene coverage when you're using this particular method, which again you see here as method 5, which is obviously a good indicator for you to be able to then use this so that we can downstream then use qPCR analysis or other methods to validate the genes that we find out. So this is all looking interesting. And in this particular case, we have, for example, with method 4, when we compare GBMs to controls, we're able to find 14 genes that are enriched in these GBMs, versus method 5, you see 82 genes that are enriched, while with a total RNA-seq, you have about 22, while genes that are depleted are 9, 13, and 0, as you can see. So method 5 clearly is outperforming all of them in terms of upregulated and downregulated genes. More specifically, if you try to overlap these genes, so GBM high would be the genes that are upregulated, or GBM low, the genes would be downregulated. So you see that about 4 of them only overlap between all 3 methods. while in the low expressed genes, none of them actually happen to overlap between all the methods. So what we did here, again, to summarize, we basically started with this first cohort of samples, 8 samples, and then we did a second validation with another 8 cohorts. So the same, different types of different patients. And the first cohort, we found these 98 genes that mRNAs that are differentially expressed between the GBMs and controls. And 83 enriched and 15 were decreased. The correlation between the samples on the differentially expressed detected mRNA is separated quite well between these two populations. And we were able, these same 98 genes, we were actually able to apply them to a second cohort of patients. And in the same method, we were able to differentiate these two groups. And currently, we are trying to do that in a larger population to see if we can do that in parallel. So this is a visualization of what that separation looks like, and those 98 genes that are differentially expressed. This is also further validation of these differentially expressed genes, where in the same 8 samples, and you can see a nice separation using PCA and a decent sensitivity and specificity on the first and second cohort. And again, using random forest feature selection algorithm, we were able to reduce these genes from 95 to down to 4 that could separate these populations even at a higher sensitivity, and we're able to do that in at least 2 different cohorts. So what we're doing now, as I said, we are trying to do that in a larger cohort, but also in parallel, we're really going back to the data and trying to find, so out of these top 4 or 5 performing genes that we're working with, where can we design primers to look at them? And that's the challenge, obviously, for all of us. But in this case, because we've been able to reduce the number of genes that are significant, we're able to say, well, why don't we go back to the coverage and actually look at where the information is. And in this case, it worked out well because the top 5 performing genes actually have similar coverage among the different 8 GBMs and 8 controls that we were looking at. So again, just to conclude in this case, so in this example, so we started with, we have this hybrid capture platform that works quite well with plasma, EV, RNA. We have 8 samples to begin with that are age and sex matched. And then we were able to have a nice separation of these, of the gene expression on PCA and identify these 98 genes. These were again validated, and we, in a second cohort, completely independent, and we were able to again confirm this separation. Right now, these genes actually, we're doing still more sequencing. We're not at the PCR stage yet. We are currently using 32 samples from our biobank. I'd say at least more than 10 of them are patients that actually have a suspected mass, but not necessarily a GBM. It could be an infectious, could be MS, could be anything. And about 2/3 of them are glioma specific samples. And then 16 of these will also have recurring disease. And so we'll validate that at the first time diagnosis as well as at the recurring disease time point. And in parallel, we'll also be doing the, hopefully these 5 genes do return, and we're able to validate them with qPCR. So another example that I was going to share with you is the Dacomitinib study. So in this case, it's these were serum samples actually. This is total RNA sequencing. It's 14 samples. These patients were enrolled in a clinical trial, and they have they are all EGFR amplified, so it's a very unique group of patient population. So they're quite homogeneous. And in this clinical trial, there were only 7 responders out of over 60 that were enrolled. And we, at the same time, we selected 7 that were non-responders. They were, we isolated the EVs from serum, and we performed RNA sequencing. Again, this is QC just showing that we get with ribosomal RNA depletion, we get still obviously quite high reads, but we have good amounts of protein coding regions. And this is just showing you two different types of normalization, the above square here and below down here. And this is on the right side, you can see the protein coding regions that we find using this total RNA sequencing method, which is about 10,000 transcripts. And it's higher than what it's been previously reported. So we're able to get a good amount of protein coding genes in this case. The interesting thing that we did here was actually to look at how can we normalize these two, how can we normalize this study? Again, thinking of validation and downstream validation, basically in larger cohorts, right? And so in this case, again, with a lot of work from Rob Kitchen, we were able to, we went back and forth many times, and we were able to compare the different type of normalization. So, and we turns out that if we actually use just GAPDH, we're still able to see most of the genes that were shown to be significantly enriched or differentially expressed in the two different groups, in responders versus non-responders or controls. A lot of these genes overlap. So we did stick with GAPDH normalization, and with the idea, again, being that we can use this type of normalization also in qPCR if we stick to GAPDH. So in this case, we were able to separate the healthy controls here from the GBM samples. So this is including all of the GBM samples that we analyzed. So we were able to break down the type of genes in genes that belong to different categories, like gliogenesis, apoptosis, and et cetera, as you can see them here. So this is just first-time diagnosis signature detection. And these were the top 3 performing genes between the two populations. Again, this is just healthy and GBM, so no treatment yet. And again, all of this is normalized to GAPDH, as you can see here. And further down, if we just look at pre-treatment versus post-treatment, we're also able to separate these two groups and quite distinctively. And you can see also here the non-responders versus responders. Again, the way we classify the non-responders and responders is by a 6-month progression-free survival. So if they made it to 6 months, that they're classified as responders. Most of the patients, unfortunately, they only make it to maybe 2 or 3. So that's really the majority of the group. And these are the genes, again, that are differentially expressed or enriched in the non-responders versus responders, again, using GAPDH. And similarly, we can look at the responders and non-responders. These are the genes that were typically telling us within the group of responders or non-responders, the changes would happen in these small group of genes. So as in non-responders, if they were high in post-treatment, so they basically correlated quite well with the group. I think that, and again, with the GAPDH here normalization, this was the one gene that correlated with the response to therapy. So being low in this case, this gene was quite low in the beginning in the non-responders, and it actually was increased after treatment. And so in conclusion, so we have an RNA signature basically to determine the presence, but also stratify these patients and assess their response to therapy in these patients with the recurrent EGFR amplified that were undergoing therapy with Dacomitinib. And I want to thank the team working on this and Exosome Diagnostics, and who's been heavily involved with these sequencing platforms. And thank you for your attention.